I want to get your take on, and Erin, perhaps we could start with you. What indications or disease areas do you believe are most ripe for successful iPSC therapy translation in, say, the next 3 to 5 years? Are you seeing it kind of as all of cell and gene therapy with a focus on or a progress in non-oncology indications? And I should point out that our audience has indicated that 41 over 41% have noted that they believe ophthalmology is in the near term has the greatest potential for iPSC derived therapy. So wondering what you think about that.
Yeah, thanks. I mean, I think I appreciate the ophthalmology voting on that, because we're certainly at Astellas, we have our clinical stage RPE program for geographic atrophy secondary to AMD. That's our flagship program, and we just recently shared some interim data from our ongoing clinical trial just before the ARVO conference, the Retinal Innovation Summit earlier this month. But I think in addition, there's really exciting work coming out from Blue Rock as their dopaminergic neuron for Parkinson is heading into phase 3. And then similarly, Vertex's islet program is moving into phase 3 as well. So I think those three areas, brain, pancreas, and eye are the front runners in the non-oncology space. And it's, the field has been leveraging lessons learned, and, maybe it isn't moving as fast as some originally hoped, but I think the progression of these trials to phase 3, particularly for Parkinson and type 1 diabetes, is really encouraging, and I think that's going to be a huge improvement and advantage for maintaining that excitement over the next 3 to 5 years.
Yeah, I would agree, and I'm certainly happy to hear it. Julie, what's your take? What do you think are the indications or disease areas that are perhaps most ripe for successful iPSC therapy?
Yeah, I would echo Erin on the top three: neurodegenerative, even Parkinson's and Alzheimer's opportunities. But overall, I think cardiovascular diseases could be included in that after the top three, and possibly even musculoskeletal disorders. So I think that there's opportunities where cell replacement is required. But I think Erin hit kind of the top three that we see where investors are really interested in looking at that type of technology.
Sure. Julie, so while I have you, how are advances in gene editing, so for example, CRISPR, influencing your approach to designing optimizing iPSC-based therapies?
I would say the big piece with gene editing right now, whether it's CRISPR or MAD7, is really looking at the opportunities to be immune-cloaked. We are involved in some work looking at tissue-engineered organs and being able to repopulate that with cells that would not require immune suppression. So I would say that's just a super exciting area, and for many of these diseases where we can knock out HLA class 1 and class 2 and then knock in other such as CD47 or HLA-E or a modified F that's going to help to sustain those cells without the need for immune suppression. So I think, targeted knockouts and overexpressions are really have significant interest right now, allowing different opportunities to be treated without this lifelong immune suppression. Besides the fact that it's, a personalized medicine and we can specifically tailor that cell for the patient. So I think these genetic editing influence the design and the optimization of these cells. So, and then looking at safety improvements, of these cells. So whether that's just doing the gene editing or, as I described, coupling that with other technologies that might reduce the number of gene edits that we're looking at. But I think it's making a huge difference, and it will in the future, hopefully, where we won't have patients on, a handful of these drugs every day.
Sure, sure. And Erin, before we continue on to our next topic, I just want to, of course, give you the opportunity to chime in on any advances in gene editing influencing the designing or optimizing of iPSC-based therapies.
Yeah, no, I think Julie did a great job of answering this question. Certainly, the immune cloaking or immune evasion is a huge use for gene editing technologies. I think it will really enable us to make these PSC-based therapies available for a broader range of patients if we can administer them and not have to have them, patients on immunosuppressive therapy. That's just it's such a game changer and helps us realize the off-the-shelf potential. Yeah, and as Julie mentioned, there's a lot of different approaches to this, variations on the theme of what to, the HLA class 1 and class 2, but what other tolerogenic protein should we be expressing to avoid NK-mediated attack, or to help with immune evasion in the long run. And then, of course, engineering in a suicide gene as a safety switch so that you can get rid of the cells if anything untoward were to happen. But, I think as the field moves forward with this, it may not be a one-size-fits-all. There could be the right approach may depend on the number of cells and the location within the body where the cells are being administered. But certainly a lot of excitement around this, and looking forward to seeing more of the clinical data on how best we deal with the topic of immune evasion and how to make that a reality.
Just to add to what Erin said, was this also gives us an opportunity to reduce the cost of goods and hopefully see that more patients could be treated with these products. So that's really exciting for us in the field. As we know, when we look at other applications such as immunotherapy, there's probably in the US, 20% of the patients are treated that can be treated. So, what's happening to the other 80%? And we really need to figure out how we can reduce the cost of goods so that these more patients can be treated. And that's only one issue for the market access.